for the early session here. I'd like to thank the organizers for inviting me to give this talk and the awards committee for helping me get here to give it. And as was mentioned, I'll be talking about some compounds that we've been working on the structure elucidation. They're anti-quorum sensing against Staphylococcus aureus. Our lab is focused on methicillin-resistant Staph aureus, or MRSA, MRSA. This has become a very large problem worldwide. This is some numbers from the CDC a couple of years ago. Over 80,000 infections, over 11,000 deaths. This is the hospital-associated type. So these are the ones, these are the types of infections you get after surgery or when you're sick. These generally affect immune-compromised in some way patients. The bigger issue that's arisen lately is the community-associated Staph aureus. And this is a type of staph that infects healthy individuals. It's much more virulent, produces much more toxins, and can actually kill healthy individuals. It's not actually the staph infection that causes the damage. They produce a whole host of exotoxins that do damage to the skin and soft tissue, and this is the lose your breakfast slide number 1. There's more to come, I give you a warning. So toxic shock syndrome, this is the syndrome that you see warned about on the side of every tampon box since like the late 70s. It's a systemic reaction to the toxins. It can be fatal very quickly if not caught early. Scalded skin syndrome, this mainly affects babies and neonates. So young children, very young children, and it causes their skin to literally just peel off in sheets. It's very painful, I'm betting, and can be deadly very quickly because they're losing a large surface area of their skin. And the third one up there is kind of the catch-all. This is abscesses, necrosis, and sepsis. These are from hemolytic toxins. And these are the lesions and the abscesses, the big pussy, nasty-looking sores that you see associated with a staph infection. Again, these can get out of control very quickly, cause some very nasty damage, permanent scarring, and ultimately you can die if you don't treat this rather quickly. And as I mentioned in the title, we're doing quorum quenching. This is not antibacterial, these are non-biocidal. We are looking to shut down this accessory gene regulator system, there's your pointer, or the AGR system that is responsible for quorum sensing. And quorum sensing has to do with interbacterial communication. One staph on its own, a little staph bug floating around, it's not terrible, right? What happens is when they get together, they send out these chemical messengers telling other staph to upregulate their toxin production. So you have a chemical messenger coming out and being picked up by other ones, and the virulence exponentially increases when they reach quorum. And as we all know, bacteria multiply rapidly, so you have this just massive spike in the toxin production. What we're aiming to do is find compounds that prevent this from happening, so they turn off this chemical messaging system, either preventing the chemicals from being excreted or shutting down the regulator or the receptor on the, so that they can't receive the signal and upregulate and produce more toxin. The plant that I'm focused on is Kseniya sativa. This is the European chestnut. You know, chestnuts roasting on an open fire, the standard food stuff, right? Everybody has at Christmas. Traditionally in South Italy, this is used as a skin wash. They make a decoction of it. They either take a bath or wash themselves for skin inflammation. But they're using the leaves there. So it's a pretty common food crop, but as far as being used as a decoction for this type of thing, it's not very common. So this was our lead. We do an ethnobotanical approach in our lab. So just to kind of give you a full overview, I'm not sure we've got lots of different parts of that type of research at this conference. So starting at the beginning, all of our leads are identified by field interviews. These are of course done with prior informed consent and an agreement, a benefit sharing agreement already in place. Voucher specimens are taken of all of the relevant plants. Those vouchers are deposited locally in Italy at one of the herbariums there, as well as brought back and deposited at Emory University's herbarium. Bulk specimens are collected, they're dried in the field, and then of course mailed back to the lab. They're ground in a Wiley mill, and then we do a 95% methanol couple maceration as our standard extraction protocol. We dry it down, in vacuo, resuspend it, and freeze dry it to get a nice powdered extract. That extract is then screened using a quorum sensing assay, and all the fruit extracts are looked at, and the most useful lead is identified. In this case, it's Kseniya sativa, which is then partitioned with a cuption scheme which is hexane, ethyl acetate, butanol, and water. It's a pretty standard natural products partitioning scheme. All of those partitions have been tested in the same bioassay, and we identified the ethyl acetate partition as being the most active partition, so that underwent various steps of chromatography, open column plus flash chromatography over normal phase, repeat that a few times, and ultimately do a reverse phase semi-prep to end up with a very active fraction that's engaged at the end. So how do we know this is a quorum sensing or quorum inhibiting compound? This is all 4 AGR allele types. If you look at this nice horizontal line at the top, these are the growth curves. So you don't see that dipping down, so that means that the staph is growing. We're not killing off the staph here. But what you do see is these plunging curves with all 4 of the allele types that are dose dependent. So that is the fluorescence associated with the YFP marker tagged on each of these alleles. So we're showing that as we increase the concentration of our extract being applied, you're dose dependently shutting down the production of these exotoxins. So again, this is not killing the staph, it's not bactericidal, this is a quorum quenching botanical extract. Again, this is, sorry, breakfast losing slide number 2. It's the last one, I promise. We took our extract and put it into a murine model, a mouse model. It was co-injected with a high load of MRSA intradermally, and then at 2, and then again at 6 days, we looked at the lesions. So this is just the vehicle. You can see it's a really nasty lesion that forms if there's no treatment. And then dose dependently, low concentration, high concentration of the botanical extract, you can see that the lesion is not even there after 6 days. This is a control, it doesn't produce delta toxin, so you still have the infection, but you don't have the damage in the lesion formation. So we have an effective compound. Is this effective botanical extract safe? The answer is yes. So again, in the same mouse model, vehicle and the extract intradermally injected, but there's no staph here. You don't see the formation of any irritation or lesions, so it's a safe treatment, at least in the mouse model. So what's it look like? We have something that's effective, we have something that's safe. This is the HPLC chromatogram. This is the crude extract, the ethyl acetate partition, and one of the most active initial fraction, 224 CF2. You can see an enrichment of the compounds around 60 minutes here, and these are the compounds that are responsible for the bioactivity. We go in and look at an LC-MS chromatogram. This is a positive ESI of 224 CF2, again, that fraction. You've got 94 compounds in there. So again, it's still very complex. It turns out a lot of the compounds don't have chromophore, so it looks really nice in an HPLC, but with an MS, you end up with something that looks more like this. Through further fractionation, we identified the numbers that are in red. So these are methane peaks, compounds that are actually responsible for the activity so far. We still need to determine if it's a single compound or a synergistic interaction of all of these, but we've got it down to 11. And of those 11, a literature search was performed based on the mass spec, and we came up with a nice library of potential hits. These are the ursine and oleine derivatives. The ursine has a single methyl at this position here. All the other ones are oleines. They have this 2 methyl groups at the top position. And then we have a whole host of non-pentacyclic triterpenoids as well. We've got 1 steroid possibility, a couple of lactic acid possibility, and the rest of these are all tannins. I promise that's the chemistry structure of the slide. Everybody can wake back up. What I'd like to focus on out of all of those structures are these 2 common core structures. So you have this ursine and oleine, as I mentioned, they differ only in the methyl position. The double bond, it's put in there wavy because depending on which structure it is, that moves around a little bit, but there's always at least one unsaturation there. It's always an ursine or oleine derivative here, and then 1, either the acetyl group, hydroxymethyl, or the carboxylic acid. There are 2 of those on all of the core structures. So you have a ursine plus 2 of these, or an oleine plus 2 of these. Occasionally it's 2 of the same, usually it's 1 of each. So the position of these groups is going to be what's important, and we would like to get into doing structure activity studies where we're moving those groups around and seeing actually what position it is that is the most important for finding and inhibition of the system. But currently, we do know it's this core structure with 2 of these side chains bonded onto it. Also very exciting when you kind of start digging into the mass spec data is there are 3 peaks that are unreported in the genus. This is based on looking at the molecular mass. They all share this common 781 ion, and it's peak 47, 48, 49. They account for about 5.5% of the total extract composition. So this is a significant contributor to the active partition, or the active fraction. And there's nothing that matches them in the literature. There's obviously some differences in the ionization, but they all have the same parent ion, so very likely structural isomers that come out very close together that have the same parent ion. The empirical formulas are listed different here, but the difference in the C57 and the C59 is less than 3 to 5 parts per million, so it basically could be either. We're just reporting the most, the one that matches the data closest to this slide. So really exciting to have at least a new compound for the genus to report, and we're in the process of isolating that in enough quantity to do actual full unstructured elucidation. So with that, I'd like to conclude. We identified an ethnobotanical lead, Kseniya sativa, from a skin wash. It is both effective and safe in a mouse model. We also tested it against in vitro human skin cells, and it's safe in both of those models. We have 2 common core structures that are responsible either individually or in synergy for the activity, and a potential new compound to the genus that's also contributing. And we're currently isolating all of those at enough compound of all of those to go do a full NMR workup and get full structure elucidation on that. Thanks to everybody in my lab that's helped with this work. They're a great bunch to work with. The Horseville group worked with the mice and the YFP monitors, of course, our funding source, and none of this would be possible without the local community in Italy. And of course, thank all of you for getting up this early and coming to see it. If you want to find out more, this paper's been submitted. It's currently in press in PLS 1, so you can get all the details, or you can ask me questions now.